Welcome to the Good Chemistry Podcast. I'm your host, Nick Jikomes, and today's episode is supported by Demetrix, a biotechnology company doing fascinating work using cutting-edge bioengineering techniques to create yeast cells that have the ability to produce cannabinoid compounds at an industrial scale. I spoke with Jeff Ubersax, the CEO of Demetrix. Jeff has a PhD in biochemistry and molecular biology from the University of California, San Francisco, and he was a postdoc at Stanford University. Jeff and I spoke about how his background in yeast molecular genetics enabled him to transition from academic science into the biotechnology industry, and how Demetrix is using cutting-edge techniques to engineer strains of yeast that produce specific cannabinoid compounds at a large scale. Jeff talked about the science of what makes this kind of biotechnology possible and why Demetrix has chosen to focus on cannabinoids specifically. Jeff also talked about what it was like for him to go from being an academic scientist to running a biotech company. And he provided a lot of great advice for graduate students and postdocs who might be thinking about that type of transition. As always, if you enjoy the content, please do like, share, and subscribe to the podcast. We also have a Patreon account. It's under Good Chemistry or Nick Jikomes. You can search for either of those. And patrons provide a monthly donation that's as little as $5 a month, and that helps keep the podcast completely ad-free. And with that, here's my conversation with Jeff Ubersax. Jeff Ubersax, thank you for joining me.
Great to be here.
So can you start off by just telling us a little bit about your scientific background and then what you're doing now?
Sure. So I have a PhD in yeast genetics and biochemistry from the University of California in San Francisco, which is about 2 blocks away from where I am today, which is in my house in San Francisco. I've always been fascinated with, how cells do stuff and how biology can make the world a better place. So was at UCSF for a number of years for my PhD, and then went to Stanford, where I was really working at the interface between biology and how you can use kind of some of the new technology that's out there in DNA sequencing, in automation tools, and in computing tools to make the biology stuff go a lot faster. Because ultimately, I think what got me into science is that, a lot of really cool things and great things for the world can happen with science. And how do you make science go faster so that it has more impact more quickly? And so pretty quickly realized that, there was some cool science going on in industry outside of, the academic world, and knew a couple of the founders at a company called Amyris Biotechnologies, and joined them in very early 2008 as a yeast strain engineer. And there I was really interested in, how do you use yeast, which are super cool, to make products that the world needs and wants in a more sustainable way, in a more affordable way, in a way that, increases, purity, safety, reproducibility, all the kinds of stuff that you want. And so was there for a number of years, almost 10. And then one of the founders of Amyris was Jay Kiesling. He's a professor at UC Berkeley that I knew quite well. And he called me up one day and said, "Would you like to have coffee?" And I said, "Oh, this sounds fascinating. I'm not going to say no." And so I had coffee with Jay, and Jay said, "Look, I have some really interesting results coming out of our lab on how you can get yeast to make this class of compounds called cannabinoids. Think it's really a great opportunity to start a new company, and would love to have you come on board. And what do you think?" And so after a couple of weeks of agonizing, said, "Hey, this sounds like a fantastic new adventure. It's an opportunity to kind of run a company in the way I thought it should be run, and jump right into a class of, super interesting compounds that, think have, a lot of opportunity to improve people's health and wellness and make the world a better place." And that's, ultimately what, Jay was in it for, what I'm in for, and what Demetrix is about. So the company that got started was called Demetrix. We've been around since, September of 2017 at this point, and are using yeast to make rare cannabinoids for all sorts of different applications in human health and wellness. And the more broad mission of the company is really to a lot of really cool and interesting and useful compounds are made in nature, in plants and other organisms that are present in really, really tiny amounts. And that through using biotechnology and fermentation, there's opportunities to make them much more available and useful throughout the world.
So let's talk about yeast. So what are yeast and how are they normally used by humans?
Yeah, so great question. So we use baker's yeast, same yeast people use in bread making and beer making and wine making, but are also used in a huge number of other products that people use around the world today. So yeast are these extremely cool microorganisms that people have been working with for over 100 years at this point. And so are really their metabolism and genetics and genome and all that sort of stuff and the tools for editing them and getting them to do new things are quite sophisticated and advanced. And so have been a focus of a number of companies, including ours, for how do you put DNA from other organisms, right? This is one of the coolest things about biology to me, was that DNA is universal. So DNA is like, people will call it the blueprints of life, right? It's really a string of letters of A, T, G's, and C's that basically tell every organism how to do the things that it does, right? And the cool thing about that is that you can figure out what parts of that DNA do different things, and then you can basically get that out of the organism. Or today you can just order it. You submit an electronic file to somebody like Twist or other companies, and they'll send you that string of DNA. And you can put it into other organisms like yeast, and now those yeast do the thing that the plant used to do. And so, to give people a sense, like, on the cannabinoid side, yeast are tiny. So, some of the most beautiful structures that we've seen out of the plant world are, in my opinion, at least, are trichomes, right? They're these, beautiful kind of stalks that have this beautiful globular head at the top, and they're full of oils and all sorts of, useful products that plants make, not just in cannabinoids, but a lot of plant products end up in the trichomes. And you can fit about 1,000 yeast cells in every one of those trichomes, right? And so one of the challenges with getting a lot of these rare natural products out of plants is that you need to grow a lot of that plant biomass to actually get enough of that product to actually do stuff with, even to study it, to figure out what it does. And you can imagine if you can take yeast that where thousands of them could fit in a single trichome, all of a sudden you have an opportunity now to make a lot more of that product and make it a lot more kind of available for all the things that need to happen to really understand what they do and how they can be helpful to the world.
Interesting. So yeast are single-celled microorganisms.
Yep. And basically their job is in the world, and they've been used for 6,000 years or more, to basically, you feed them sugar and they produce carbon dioxide and alcohol, and that's how people have used them for a really long time. And they also make more cells. And so what we do basically is change some of the DNA or put new things in there and modify some of the DNA that's in the yeast cell to basically now take sugar and not make ethanol, but make new products, right? And secrete them into, the fermentation media that we grow them in, which is just, like a nutritional broth. And then you can purify the product out of there at the end of the day, and, in theory, be able to have, higher purity products, more reproducible products, because you're not dealing with some of the, other weather and rain and soil conditions of agriculture. So you can, have more reproducible, more pure product at the end of the day.
So the idea is yeast naturally eat sugar and they produce ethanol as a waste product. And we sort of just take that waste product, the ethanol, and we use it to make alcohol.
For beer and wine, or you use the carbon dioxide, right, for your rising bread, right, as an example. Yep.
And so you're saying that you guys can engineer yeast so that instead of producing ethanol and CO2 as a waste product, they produce whatever you want, basically?
That's the idea. And so, that sounds super easy. It's actually really, really hard. But basically, if you can understand how other organisms like plants or other microbes make products, then, what are the DNA parts that they need to, instruct their cells to make those products. If you can identify what those DNA sequences are, you can recode them and put them into your yeast cell. And now that yeast cell will start making some of that product, right? Usually it starts at a very tiny amount, and it requires, a bunch of work and sometimes years of work to really get them to start making kind of commercially, interesting amounts of that product, right? So
I see.
At this point, the world has probably made 1,000 different compounds that plants make in yeast, right? But very few of them are not, necessarily at the scale that you would want them to be at to make them kind of commercially relevant.
Yeah.
But that's really what there's a whole industry out there that some people would call synthetic biology or industrial biotechnology that's really aiming at taking, those yeast cells and making it so it's making tiny amounts like milligrams to how do you get them to start making metric tons per year, which is what is really kind of necessary to make have the impact on people in the world that you kind of need to have.
I see. So where the field is at, if I'm understanding, is yeast cells. So yeast geneticists can basically engineer yeast to produce any compound, more or less, but they're not necessarily going to produce that compound at a level, at a scale that you want it to. Yep. So all the work is at how do you get the yeast to produce a lot of something?
Yeah, and there's lots of things underneath that, but and what I'd say to kind of put a little bit more nuance on it is that there's certain types of chemistry that people like synthetic organic chemistry is really good at. And there's another type of chemistry that biology is really good at. And the type of chemistry that biology does is oftentimes very hard for what the synthetic organic chemists do, right? And so what most people are doing are trying to, harness that chemistry of cells and the chemistry of life, you might call it, to make, the products that are coming from other natural organisms like plants, right? Or other microbes, like antibiotics is another good example where, people or insulin is another good example, right? Where people have, those are molecules that would be very expensive to make through synthetic organic chemistry, but that, organisms in fermentation can do really well because the chemistry is just really fits, the chemistry of the cell really fits well into making those types of products.
And so are you guys focused on cannabinoids specifically?
That's where we're starting. Yep. And, I think that there's just, it feels to me like a once in a kind of career opportunity with cannabinoids, because there's kind of been a convergence of regulatory changes as well as, some of the science that's happened to understand how do plants make these products. And the combination of those two things have really opened up, a potential for a very, interesting class of natural products that haven't been really available or well studied to be, to study them, to say, what can they do? How are they different? And so I should step back and say, one of the things that we're really focused on is the rare cannabinoids, right? So the major cannabinoids most people are familiar with is THC and CBD. The plant makes, more than 100 other cannabinoids that haven't been studied nearly as much, but that because they've been present at such small amounts in the plant. So what we've done is, basically made some of those and then, are working to understand, okay, what do they do? What does their safety profiles look like? What do their efficacy profiles look like? How can we use these things in everything from, skincare products to foods and beverages to supplements to even pharmaceutical ingredients to, improve people's lives?
Interesting. So before we get into the specifics on the cannabinoids, one more question about the basic biology I have and what I presume is an engineering problem with this whole area, or an engineering challenge that you have to get around. So the plants have evolved to produce these cannabinoids for their own purposes. They put them, or they secrete them from the cells at the top of the trichomes, and my understanding is they're actually purposefully away from the other plant cells because these compounds can be toxic actually. So do the yeast tolerate them well, or do you have to engineer the yeast to play well with the cannabinoids?
Yeah, so it's a bit of everything, right? So that's a great question, actually. So, a lot of the work that we do is to, do two things. One is to kind of make the plant parts, like the enzymes and proteins from the plant that we're using in yeast, work better in the yeast environment, right? Because the yeast looks very different than the tip of a trichome, right? It's just a very different, it's an oily thing versus, a whole cell in a fermentation media. The second part is that we're also trying to engineer the yeast cell to look a little bit more like the trichome, right? Like the plant cell, so that, you kind of then are approaching the problem from both ends to then make enough product that it's commercially relevant. And one of the challenges that you say is sometimes those products are toxic to the yeast cell. I think, the nice thing about cannabinoids is they're not super toxic to the yeast cell. And even if there are certain types of, fermentation processes that you can put in place that help, relieve that toxicity. And you can also evolve yeast cells to be more resistant to whatever toxicity, those compounds are causing. The one thing I'll say with cannabinoids in particular is they're not particularly super soluble in water. So you don't necessarily, in the fermentations, you don't necessarily build up a whole lot of kind of like soluble cannabinoid. It tends to kind of, aggregate out of the fermentation broth and be with the cells in a way that's a lot less toxic likely.
And so why did you guys pick yeast? Is it just because of the genetic toolkit that's available for yeast, or could you do this in other microbes?
So there are other ways to do it. There are other organisms that people are working with. The reason we've chosen yeast is a couple reasons. One is, the tool set that has been developed over the decades is really outstanding, and it makes the engineering part really, really a lot more easy than it was, even 10 or 20 years ago, and much easier than it is for some of these other organisms. The second part is that the type of chemistry that kind of has to go into making cannabinoids are things that other people have had success doing in yeast, right? So the class of enzymes that are the plant uses to make these products have been, similar types of enzymes have been used in yeast to make similar types of products. And so, there are other people that are doing it in E. coli, which is bacteria. There's other people doing it in algae. And, our bet is on yeast. And we think that because of the prior success in the toolkit in yeast, it's a great choice for going forward with this product or these types of products.
So which cannabinoids have you guys started making first?
Yep. So the first one we made is kind of it's called cannabigerol or CBG. And so the plant actually makes CBGA, the acid form of that. And that's actually what the yeast makes. And then you can get it decarboxylated. So we're making, in some ways, we're making both CBGA and CBG. I think that, beyond that, we've got other products that are kind of in the development pipeline. And those are, other minor cannabinoids where, I think where we see them to be different than CBG and providing different types of, effects on or impacts on people and what different applications they might be useful in, so that we're not necessarily just making, another CBG like molecule that does the same thing, because that's not actually what the world needs. The world, we should be exploring the diversity of the plant and finding the parts that are doing, different things for people, so that we're bringing, the most benefit out of the plant into people's lives is kind of how we think about it.
And what do we know about CBG and CBGA at this point?
Yep. So not as much as the world probably would like or would need. And so, this is the issue with a lot of these rare cannabinoids is that they just, first, because the plant was a scheduled substance for so long, it was very hard to do some of the fundamental science of what do these things do. The second part is that it's very hard to get access to a lot of the rare cannabinoids because they aren't there's not much of them in the plant. And the chemistry to make them is hard and expensive, like the human chemistry, right? The synthetic organic chemistry is hard and expensive to do. And so, I think it's only in the last couple of years with kind of some of the changes on the regulatory front with the rare cannabinoids that are non-psychoactive that, there's an opportunity to use a combination of biology and chemistry to really start exploring what all these other cannabinoids kind of do, right? And so we've invested a lot of our own time and resources in doing the type of actual hardcore science that you need to do to understand what these things are going to do in your body and on your body, right? And so what are their safety profiles look like? What are the efficacy, where might they be useful in different types of products? And so some of these things are things like G protein coupled receptor assays or ion channel assays to figure out, what type of receptors are these things acting on in your body, and how are different cannabinoids hitting different receptors in different ways, so that you can then, and that gives you both a sense of what their, safety profile looks like, because there's certain, receptors in your body that you definitely don't want to be touching because they'll do really bad things. And there's other places where you'd say, okay, this is, looks like it's hitting a bunch of, receptors that might be useful in things like inflammation, or in things like, anti-inflammatory is one of the big areas that we're looking at, but there's a whole bunch of other ones as well that we've been investigating.
So you guys are doing like the receptor binding assays and all that stuff as well?
Yep. Because, our philosophy, like, I think that's really, sometimes you'll hear people talk about like the cannabinoid space is kind of like the wild, wild west. And, to some extent that's true. And what's driving that is that there's not really a good understanding of what these things do and what they can do. And the science, our philosophy is like, we need to be using the science to understand what these things are, what they are good for, that they're safe, to be able to have the impacts that we think that we want to bring to the world and the world ultimately needs, right? So, and interestingly enough, it's also very consistent with what, a lot of the regulators of this space want to see as well. So we work very closely with the FDA, as an example, to have, to help them understand, what we're doing, and help them understand how, these products, like cannabinoids in particular, can be regulated under existing laws for how, and regulatory frameworks for how, natural products make their way into all sorts of products, whether it's consumer products or pharmaceutical ingredients. And, they are super important organizations, right? Because they're there to protect human health and wellness. And to, they've been very excited to see people actually doing the hardcore science and willing to work with them through their existing frameworks to do all the stuff that needs to be done to show these things are first safe for skincare applications, like topical applications, and then ultimately ingestibles, whether that's in the pharmaceutical side, food and beverage, or on, kind of the supplement side. They're excited to see people actually willing to engage with them and do that hard work to, really understand what these things do and what they're good for.
And so because you guys are focused on the minor cannabinoids and you're not focused on THC, does that mean that you're largely free of the red tape that would come from doing anything with a Schedule 1 substance?
Yeah. So, and we started in 2017, kind of before the Farm Bill. And so we also have a registration with the DEA, right, to work with Schedule 1 substances, because a lot of these things were Schedule 1 substances a while back. And yeah, I think that, the change in the regulatory has been with the Farm Bill, that if you're making, cannabinoids or phytocannabinoids that are, have a THC content less than 0.3%, then you're falling outside of the Schedule 1 substances aspect of, of THC and cannabis. And so it does free up a lot of that. It's just much easier to work with them and be regulated as other types of ingredients would be regulated through the FDA.
And to the extent that you can talk about it, what other cannabinoids have you started to look at? You mentioned, and I really like this strategy, you mentioned, starting with CBG, but then also specifically trying to look at cannabinoids that are very likely to have relatively distinct pharmacological properties. Have you found any interesting?
Yeah, so I'll speak in general terms first. So, the way that most of these molecules have an impact on your body is that they, bind different receptors or other proteins in your cells. And, what kind of dictates which of those things they're binding to is the shape of the molecule, right? So in general, we look at kind of different scaffolds that cannabinoids make, right? So there's like the kind of natural endocannabinoid scaffold, which is like these long chain kind of loopy things. And then there's, CBG, which has like a phenolic ring and then two tails that hang off. And then there's, CBD and THC, which are, ring structures that are sometimes double rings. and then there's CBC as well, which has, another type of double ring structure. And those we kind of classify as different scaffolds, right? And so those different scaffolds are a big part of what drives how they interact differently in your body, right? So when we were looking at it, a couple of years back, we said, look, what are the kind of different scaffolds that we should be thinking about, and how do we, think that we might be able to approach making them through, fermentation and biotechnology, and then, test them to see what they're good and different for. So the structures is part of what we use in deciding to go after the products, but ultimately we make them and then we test them in some of these more functional assays like GPCR receptors or ion channels or other kind of functional assays like in vitro functional assays, to say, how are they the same and how are they different? And that kind of drives, then we go, okay, we kind of think about, okay, how hard are these things to make? And if they're, super duper hard and they require, a couple different, 15 different enzymatic steps, and a bunch of those enzymes aren't identified from the plant on how those things are made, we say, that would be really hard for us to approach and be really expensive. So maybe we'll leave that one for later and focus on ones where, keeping the end in mind, they have to be affordable and something that we can make to have, to bring them to the market. That's kind of one of the things that we need to think about. So that's one of the first kind of filters for how we think about which ones to go after. And then after we have, that, we say, okay, and then what do they actually do? And how are they the same or different? We try to find ones that are different from each other that still have, interesting applications in human health and wellness. And that's kind of how we think about filtering it down from, 100 plus different cannabinoids in the plant to these are the product 1, 2, 3, 4 that we're going to go after.
And for, in terms of CBG or whatever one is the best optimized to be produced the most from the yeast cells, can you give us a sense for scale? How much CBG can you produce compared to, say, the plant?
That's a really good question. So let me think about the best way to answer that one for a second. So, one of the, let's dive in a little bit and talk about fermentation, because that kind of is the first place to start, right? And so what do we do with fermentation, right? So ultimately it starts with a single yeast cell, right? That you've generated through, advanced molecular genetic techniques. So you've put in, a bunch of these plant things, and you've also optimized the host cell. So the yeast cell to, as I said earlier, kind of look more like, the trichome or plant cell. And those enzymes that you put in look more like the kinds of things that yeast are used to seeing, but still do the same thing they do in the plant. So you start with a single cell, and then you have to grow that, right? A single yeast cell doesn't make a lot. It's tiny, right? It's you can't see it without a microscope. It's about 10 microns big. It's not a very big cell. And so you put it on a plate, agar dish, and it grows into a colony. A colony is, a whole bunch of cells, like 10 to the 8th cells or 10 to the 10th cells. You can then scrape that and you put it in a flask. That flask kind of grows up over time. So you give it sugar, you give it oxygen, it eats the sugar, it makes ethanol, carbon dioxide, and also more cells, right? So you keep growing it bigger and bigger and bigger. And then you reach a scale where glass flasks no longer work because you can't make, 100,000 liter flask. They just, you couldn't do it. And so then people move over to what are called fermenters or bioreactors. And these are just larger vessels that supply, air, sugar, and that's basically it, and nutrients to these cells to allow them to continue to grow and now start producing your molecule. And so, the way that this works is that in the laboratory that we have in Berkeley, we go up to about 10 liters of fermentation production, right? And, the typical way that people kind of think about how much product you're making out of these fermenters is by talking about something called titer. So titer is the grams of your product that you've made per liter of your fermentation, right? And so typically in the biotechnology space, people have brought products to the market that are anywhere from, their titers are anywhere from like 1 gram per liter up to, over 100 grams per liter, right? So a liter is about 1,000 grams. So if you're at 100 grams per liter, 10% of your fermenter is your product, right? That's a lot. There aren't actually that many products that are kind of in that space. Most of them are down in the, call it the, I don't know, like the 1 to 25 gram per liter range is typically where people would be. And so from that, you can say, okay, if I had, 1,000 liter fermenter and my titers were at 10 grams per liter, I'm making, basically 10 kilograms in that 1,000 liter fermenter. And that's just what's in the kind of fermentation broth. The next step that you have to then do is you have to purify it out, right? And that's tricky and hard, and oftentimes something that people don't think a lot about. It's like, how do you get this back out of the fermentation broth? And so that's what we call downstream purification, or DSP. And that typically has an efficiency anywhere from, call it 25% all the way up to, call it 80 or 90% in the best kind of fermentation. So if you're at, let's just say you're at 50%, that means 50% of the product that's in that fermentation broth you're able to recover. The rest kind of goes down the drain, right? And so from that 1,000 liter fermenter, if you're at 10 grams per liter, you're making, 10,000 grams, which is 10 kilograms. And then you've got 50% recovery at the end of the day. You're making about 5 kilograms per 1,000 liters, right? And so
there's two basic steps here, just to make sure I'm following. So if you were thinking analogously to the plant world, someone would be growing a bunch of plants, and that plant would be producing trichomes that produce cannabinoids. You guys are putting yeast cells into giant tubs, essentially, giving them sugar, giving them oxygen. The yeast are then growing, multiplying, kicking out a fair amount of cannabinoids as well. So the giant tubs of yeast are analogous to growing a plant, basically. And the second step is getting the cannabinoid from, purifying it from those cells, which would be analogous to doing a plant extraction.
That's right. And in the fermentations, these products, at least for this product class, are outside of the cell already, but they typically, because their solubility in water is so low, and yeast grow in water, they're associated with the kind of, because cells are, kind of lipids with carbohydrate cell walls. That's what cells or yeast cells have. So that looks more like what cannabinoid looks like. So cannabinoid kind of sticks around in that, the non-soluble phase, which includes cells and your product. And so you can basically do like a what's called a liquid solid centrifugation, where you centrifuge, which is just to spin to separate the liquid phase from the solid phase. And then you almost have something that like you think is very analogous to the plant world, which is like, we've got, plant biomass and we've got cannabinoid. How do we get that out? And so, the DSP process in many cases is with some sort of extraction, oil extraction, or, there's a whole bunch of different ways you can get it out of there that you can then do, further purification on down the line through crystallization or other types of tools.
And in the plant world, when you're growing the plants, harvesting them, and especially if you're doing extractions for making concentrated products, you often have to worry about things like residual solvents from the extraction process. You have to worry about pesticides maybe that were used to help facilitate growth of the plant. Is there anything like that you have to worry about when you're engineering these compounds to come out of yeast?
Right. So it's one of the coolest things about fermentation is, you don't have to deal with a lot of the kind of pesticides or, heavy metals or other types of stuff that come through with that. Certainly, like, organic, like residual solvents and stuff like that is something that everybody should be concerned about. That is something that anybody who's doing any type of purification process, whether it's, from, cannabis plants, hemp plants, or from, even making, pharmaceutical ingredients oftentimes use, organic extractions during, the chemical syntheses. So, residual solvents is always something that everybody's going to be paying attention to. It's something that, we pay attention to. I think the advantage of doing it from yeast is that typically, kind of like the product being made to, the biomass is just a lot greater. And you're not extracting a lot of the other things that you typically extract during like a plant extraction, like polyphenols and waxes and a whole bunch of other stuff. Like yeast just doesn't have that. And so the product that you get at the end of your purification process, when we first got into this and we started talking about cannabinoids, we were talking about, basically high purity product. And people would be like, our product is pure. And they'd be like, well, what does that mean? Because in the cannabinoid world, pure oftentimes means, greater than 90%, we might call it pure, and greater than 98 or 99%, then you have an isolate. And we're like, well, then we have an isolate. Because what we're used to in the fermentation world is, purity is typically over 98 or 99% pure. We just call it pure. And that's what we're used to calling it. So entering into the plant world where there's both this purity and isolate word was, confusing to us at first, but we really quickly adapted to that. And so we basically make product at the end of the day that's, greater than 98, 99% pure out of these fermentations.
And can you give us a sense for time? So when you're growing the plants, I'm not a grower, but you're talking about months of time to go from seed to plant to harvest. Yep. And do the whole thing. What's the time frame for this type of approach?
Yeah, so it's usually about a week to go from, that kind of single cell or colony all the way up to, hey, we've got, typically we think about when you're talking about commercial scale production, we're typically talking about 100,000 liters or so. And so to give you a sense of scale, 100,000 liter fermenter is probably about, 20 to 30 meters tall. So it's big. Yeah. It's a lot of volume. So what we get out of that at the end of the day, is hundreds of kilograms of product that you then run through the purification process. And, that end-to-end process, typically, the fermentation might take a week, and then the purification takes another week. And sometimes, that's about, then there might be shipping and logistics and all that sort of stuff, but it's generally like a 1 to 2 week process.
Wow. And what does pure cannabinoid look like?
So it depends on the cannabinoid is the answer. So like pure CBG looks like a white crystal, which is very, very cool. There's others that are hard to get crystals of. They're more oils. And so it just depends, again, on the shape and the structure of the molecule. Oftentimes, drives the chemical, what that looks like in its pure form. But CBG, I mean, that was one of the coolest things for us, is that, one of the hard things with science is that, what you do oftentimes doesn't have like a physical or tangible thing that you can touch and say, I did that, right? It's more esoteric, and it's more like, hey, I learned something new that nobody else in the world may have known before. And that's fun and rewarding in its own way. But to actually go from, a scientist, like a project where we started, from no lab in 2017, like nothing, we were working out of, somebody's dining room. And, to a point where, 3 years later, we were running, 15,000 liter fermentations and making, kilograms of product that you could actually see. They're like buckets of products that are, highly pure crystalline CBG. Like that is super rewarding to say, we actually made that. That's pretty cool.
Yeah, no, that is cool. And so there's the fermentation step, there's the purification step. You end up literally, in the case of CBG, with buckets of crystal, kilogram quantities. Where does it go from there? What types of places, what types of companies are you guys actually selling the product to? Is it people doing clinical research? Is it people making consumable products? Is it all the above?
Right. So what I'll say about that is we're not actually selling product yet, right? So
okay. You're just developing the tech?
We're developing the fermentation, the purification technology, and starting to build those commercial relationships, right? And a big part of this is understanding what we have and what it's good for, and doing all the safety trials. So actually those first kilograms of product are things that we are using to do all the types of safety studies that folks like the FDA require you to do, right? So some of these are typically when you talk about fermentation ingredients coming to the market, the way that they come to the market is through a couple different pathways with the FDA. So if you're going into food and beverages, you generally are doing what's called a generally recognized as safe notification with the FDA. And that requires, a large amount of toxicology and safety studies first, where you're doing kind of long-term studies at reasonably high doses, typically in rodents like rats, to show that, if I do 90 or 180 days of high dose treatments of these things, that those rats aren't going to be, having tumors or other types of things that really prove that these things are safe. And so that takes a significant amount of product, and is where a lot of companies would typically bring some of their first kilograms that they're making. The second part I would say is that we have gotten to the point where we're sending samples though of these products out to, and the samples range from call it a gram to hundreds of grams of product to all sorts of kind of the first applications that many people are talking about going for is topical applications in skincare and consumer products, because the regulatory threshold for those is kind of the easiest to get through. It's basically you have to do irritation of skin and eye testing as an example, right? And those are relatively straightforward short-term studies, not half a year type of studies. And so that's where we've started, and so have been prioritizing samples going out to very large global conglomerates, all the way down to smaller independent brands that are really interested in the cannabinoid space.
And can you give us a sense for like the cost effectiveness? I would imagine that this is probably, or you're aiming to make it much more cost effective just in terms of speed and literal cost to make, a kilogram of product compared to growing plants and extracting from the plant.
Right. So the way we think of cost is really about, it needs to be at a cost that makes it available and affordable for the applications that it's going into. Right. And so a lot of that's kind of driven by, the efficacy of it as well. Like, people are basically, like, at the end of the day, people are paying, if you're in the non-pharmaceutical space, so if you're in consumer products as an example, people are paying, for the effect of that thing, right? So, and they're paying, dollars per effect. Is one way to think about it. And so, most, in the consumer product space, like if you're talking about kind of like medium to high end cosmetics that might sell, call it in the $60 to $100 range, most brands are only going to pay at most like $5 of that 100 is going to be the ingredients that they use. Right. And so you've got to be able to have, be able to, generate, an effect that's positive for the consumer in that $5 kind of range, right? So that's how we think about pricing. And so, for a lot of these products, the rare ones are really hard to get from plants. CBG, there are definitely very smart, talented plant breeders out there that are, starting to, I think, make, hemp strains that are making significantly more CBG, and the price of that's coming down. And so, we've got to be competitive with that, at least, is kind of the way we think about it.
Interesting. So, any other cannabinoids you can tell us about specifically that you think are interesting that, not necessarily either ones that you guys have produced or that you're thinking about producing? Off the top of my head, you mentioned some of the big ones. So CBG, CBC, people are talking a lot about CBN, which isn't actually produced by the plant. And then there's these new ones that I've been starting to hear about that are present at very low quantities, but potentially have very high like receptor binding affinities, like THCP. Are you guys looking at any of those?
Yeah, so a lot of those other ones are kind of tinkering around like with the tail of the cannabinoid, right? You think of the two ring structures, it's got a little tail that hangs out, and like THCP is a different length tail, basically. And that does seem to impact, both the affinity for different receptors and the diversity of receptors that these things hit. So again, it's changing the shape of the molecule in a way that, I think, to us at least initially was a little bit surprising that you just change it a couple carbons at the end of that tail and you have a much tighter binding thing. But that's one of the things, biology is always full of surprises. There's a lot of stuff you don't even know you don't know. And so there's definitely a lot of interest in both inside Demetrix and in the field in general, and how other variants of, even some of the common ones like CBD, CBG, THC, how those different variants might change how they interact with your body, right? And so we've definitely, like, of the 10 plus different cannabinoids we've tested in a bunch of these assays, some of them are definitely some of those variants that have changes on that tail length. And some of those are present in really small amounts in the plant, and it makes them particularly hard to get through the agricultural kind of supply chain, and are places where we think there's real opportunity. There's another kind of flip side to this that we can also do that's allows us to make kind of what we call new to nature cannabinoids too. So one of the things that we can do with yeast fermentations is we can feed them not just sugar, but some of the other precursors to kind of the cannabinoid pathway. Some of those precursors might be things that aren't found in nature that chemistry can make, right? Human chemistry can make. And so then you end up with a cannabinoid scaffold that has, let's say it has a tail that has, some non-natural structure to it that you are able to put into it because you can feed yeast these chemical precursors that you could never feed a plant, right? So we can make new to nature cannabinoids as well that, are mostly going to have applications in the pharmaceutical space and not so much in like consumer space, but are also something that we're pretty excited about being able to do.
Wow! So you're literally engineering Frankenstein yeast, but then you can also give them precursor molecules so that you're creating brand new, never before seen cannabinoids.
Yeah, we don't like calling them Frankenstein yeast. A lot of the products, and this goes back to a great question that we often get is, I think it's really important to understand that, there are a number of products in the world today that people use that are coming from, highly engineered microbes that people have worked on for, a long time to get to make products that are, without that technology really wouldn't be available to people. So I think one of the, my favorite kind of new ones to talk about is like, they're called human milk oligosaccharides, right? So these are, sugar molecules that are found only in human mother's breast milk, right? And they're thought to, and have been shown to have a role in the developing immune system by supporting, beneficial like microbiome in the infant's gut, and then also have indications that they're also important for brain development, right? And so these are now made there's both made and being developed by some of the biggest companies out there like BASF, DSM, DuPont, and others are making these human milk oligosaccharides through fermentation and including them in infant formula to now, provide benefits to infants that used to only be available through breastfeeding and now available through formula, right? So that's a good case where, if people were saying, hey, these are Frankenstein yeast or bacteria, people would be like, oh, no way, like keep me away. But they have, they're not that scary. They're not things that, if they get out into the wild are going to take over the wild population because, basically what we're doing is, in the case of yeast, right? Yeast are normally taking sugar to make carbon dioxide, ethanol, and more cells. What our job is, as kind of like on the strain engineering side, is to make those cells make less ethanol, and make more of your product. And oftentimes that comes at the cost of them making fewer cells as well. And so the interesting thing about that is if you were to, kind of compete in a growing experiment, these engineered yeast versus a wild yeast, that wild yeast just crushes it. Like, after a couple, even days, you would not even be able to detect any of your, engineered yeast in the worst case scenario. So, and this is, I think, where a lot of these companies, including us, take their responsibility to, we're here to try to make the world a better place. We are very conscientious and work with the regulatory agencies to help them understand what we're doing, both on, what are the molecules we're making, but, how biotechnology in general is generally a very safe activity that doesn't, have dangers that if one of these things gets out, the world's going to get high on THC because there's a yeast cell out there making THC that everybody's just inhaling or whatever.
So how did you first get into yeast biology?
Oh, that's a great question. So, when I was an undergraduate student at the University of North Carolina, and I was in an undergraduate lab there that worked on these things called motor proteins. And so another really cool things in cells is that, there's tons of these little machines like enzymes, like proteins are in there. A class of them are just little machines that actually like use what's called ATP, which some people call like the current energy currency of the cell. It's just adenosine triphosphate to use the energy stored in that molecule to actually change shape and move, right? So there's like motor proteins like dynein and kinesins and other things, actin motors that actually change shape and move as they take in ATP and then make it into ADP or either ADP or AMP and release that phosphate. Those phosphates contain really high energy bonds. And I was always fascinated by how enzymes in cells like do the things that they do, and how does that work? That sounds just so cool. And so that kind of drove me to look when I was going to grad school to say, where are some of the best places in the world that kind of study those types of things? And UCSF was one of those, and got into UCSF, and you do these things called rotations where you rotate through a bunch of different labs, and try, get a feel for how that science is done in that lab and what your interests really, where they lie and everything. And so went through a couple different labs. One of them happened to be a yeast lab that was studying how do yeast cells grow and divide, and had some really cool technology for ways to kind of really start understanding that in yeast cells. And yeast was just so much fun. It's so easy to deal with compared to some of the other things. So I also worked in like a Drosophila lab, which is fruit flies. Fruit flies are really hard to take care of. It turns out, like, you've got to, feed them, and you feed them actually, like grapefruit juice with yeast blended into it. And they're hard to deal with. You can't freeze them because, if you freeze them, they die. And so you can't defreeze them. And yeast was just so convenient, and has, so many, like, there's been just a tremendous number of kind of fundamental discoveries about cancer, about all sorts of things, how biology works in these model organisms like yeast. And it just seemed like a great tool to understand how some of the science works. And so got started on that as a grad student. And then when I went to do a postdoc, we worked on both human cells and also what are called xenopus oocytes, which are frog eggs. And frog eggs are some of the biggest kind of single cells that you can actually see. They're millimeter size scale things, but they're still single cells. And human cells and other kind of higher mammalian cells are just harder to deal with than yeast too. They grow a lot slower. So a human cell divides about once a day, whereas the yeast cell divides once every 90 minutes. And so you can just do a lot more with yeast. And so then I heard about, through some grad school folks connections and postdoc connections, that there's, companies out there in the world that are actually using yeast to make products that can make the world a better place. I was like, that sounds like a lot of fun. I should go see what that's about, and checked it out and was really quickly hooked. And so this is fun. This is what I want to do.
So it was actually in graduate school that you started to understand that you were interested in industry.
It was more, it was a mix of grad school and postdocs. Grad school at the time, was, and this was, a thing that I think is still evolving. There's a lot of, at the time, it was focused on training you to be, a professor. And, some of the alternative careers that people might pursue, whether it's like law or industry or science policy or a whole bunch of other things, were a lot less emphasized than they are today, which I think is a great thing. And it really became when I was a postdoc, kind of seeing, when you're a postdoc, you're doing an independent research project in somebody else's lab. And usually the path there is that you then, start your own lab at a different university doing some of that work. And just seeing, the job searches and how competitive it was, we would see, 250 applicants for a single, position as a professor. And, then you get down to 10 people that are interviewing and one person who's actually getting the job. And it was also, also a bit, isolating in academia, because it's like, it's your project, it's your thing. There are other people around you that are working on similar stuff, but not identical. And then I got, kind of a friend, a postdoc in the lab, his name was Zach Serber, went and joined Amyris because he knew some of the founders there really, really well. And they kind of enticed him. And I said, he was like, you should come too. And I said, well, you go try it out and see how it is. And I think that was like in September of 2007. And, by October and November, I interviewed at Amyris and was like, yeah, this sounds like a fun thing to try. So it was a good opportunity to, see how, and I think the thing that most people found as they move into industry is it's not your project. It's the project of, 10s, 20s, or even 100 people that are working on the same thing. And that makes the science go a lot faster, and it's a lot more collaborative, and there's a lot more kind of idea exchange. And so it's also sometimes more applied. And so the stuff, like the output of all that at the end of the day is like real tangible benefits to people, which, basic science has real tangible benefits too, but they're hard to touch. They're not as tangible, right? So a lot of people find that really rewarding, and myself included.
Mm-hmm. You're the CEO of
Yep, I'm the CEO of Demetrix.
So, you have the scientific background, and then you spent a number of years in the private sector. So that's sort of your two-pronged training for being a CEO. Did you ever imagine yourself becoming someone like this? Or do you think does it feel weird being a scientist running a company, or is it actually a natural fit in some ways?
So a bit of all of that, actually. So, I think that there are, certainly the job I have today is very different than I necessarily thought it would be, right? Mm-hmm. And some of the personal challenges of being, I think, a scientist CEO is that you don't actually get to do a lot of the science anymore. Mm-hmm. Like I haven't been in the lab in a really long time, and people in the lab probably shouldn't let me in the lab at this point because I don't know where anything is, or so I kind of, that's different, but and to a certain degree, being able to do some of that lab work and getting excited about, hey, I set this experiment up, tomorrow I get to see the results, is something that I definitely miss. Mm-hmm. Right. But the parts that I think are exciting is that, you're also like, as a CEO or even a senior leader in the company, you're able to, a lot of the hard parts of getting science in industry done is actually related to, how do you get teams of people to work together and then across different teams to drive the science and technology forward as fast as possible. Mm-hmm. And the people aspect of that was just fascinating to me. Like mm-hmm. people, each person's an individual puzzle. Like how do you best, get them, to do their best work and get them to be able to do things that they never even thought that they could do. Mm-hmm. Like that side of it kind of really drew me towards like, hey, the people side of this stuff is just as interesting and oftentimes just as hard as the science that we're mm-hmm. trying to do. Mm-hmm. And that a lot of what, I do now is like, organization and team and how do you get the people working together, and how do you, say connect what they're doing in the lab to, the impacts that they're having on the broader business. Like that stuff is hard and challenging and fun. Mm-hmm. And so that part's, really rewarding and very different than I imagined doing.
I think that's the answer to what my next question was going to be, which is, what's the most interesting, so much of science is driven by what you're interested in. I was going to ask you, what is the most interesting part of your job that's not the science?
That's not the science. Yeah. I mean, boy, there's a lot of interesting things. I think that especially being in the cannabinoid space, mm-hmm. there is just like, I find it so exciting to try to, to be able to talk about the science of, what we do and also what cannabinoids are and what they can do. And, there's a lot that's not known and that there's stuff that we're starting to understand. There's stuff the world's starting to understand about what these things are good for and how they're going to have positive impacts for people. Talking about that stuff and both, so helping, talk about that and educate regulators, investors, consumers, big CPG companies, like all that sort of stuff is fun. Like ability to talk about science in a way that people can kind of grasp some of the basics that aren't necessarily scientists and see mm-hmm. why it's cool and why it's exciting and get people excited about that is also something that's super fun and rewarding.
Interesting. So what, do you have any general advice that you would give to like graduate students or postdocs who are in academia that might be thinking about moving into the biotech world or anything like that?
Yep. So my first thing would be to try it. So there are a lot of universities now are starting to have like, industry internships that they mm-hmm. provide for grad students and sometimes postdocs too. That is a fantastic opportunity to try it and get a taste of what it's like. And so, I would encourage people to, the things that I think get most people hooked on the science that goes on in industry or in industry positions in general is, that um, it's a lot less of, and this is also a big transition that people have to make. It's not like your project anymore. It's like it's like there's 5 or 10 or 100 people that are working on the same thing, and that makes the science go a lot faster. But it also means that you've got to let go of this feeling like this is my yeast strain or this is my experiment. It's not. There's lots of people that touch that now, and so it's like the things that I do have a lot more kind of impact, which is both good and also something that people need to learn about. And so experiencing what industry life is like is kind of the best way to get a taste for that and to understand if that's what you want to do. There are so many other places where scientists can have impact in the world, though, that I would also encourage each individual person to think about what are the types of things I'm interested in. And if I don't even know, ask people about what are the places that I can go after graduate school or after a postdoc, because they range from going to Washington D.C. and doing science policy to going to a law firm or going to law school and talk about intellectual property or other types of law. There's just so many different applications of the training that you get as a grad student on how to think analytically and scientifically about problems that you can have impacts in all sorts of places. And the more you try, the more likely you're to find the place that you find was the right spot for you.
Speaking of IP, I imagine a lot of the value for a company like this comes from the IP you guys are generating. And I would guess that a lot of that has to do with the methods you're developing for how a particular yeast strain can be engineered to tolerate and produce a particular type of compound. Is that accurate?
Yeah, so there's kind of like 3 big buckets for how to think about IP from this type of company. So one is the individual enzymes and pathways that you use to make these products in the cells, right? So that's kind of one level. The second level is kind of like methods of production. So there's like fermentation based IP, there's downstream purification based IP, and then the last bucket is kind of like the applications IP. So once you have a product, what can you use it for? And so I think everybody, all biotech companies are valued to a certain degree off their intellectual property, and it's a big focus of us and others to think about all 3 of those buckets.
And then can you take us back, you touched on it a little bit, back to the origin story for the company. So another scientist invites you out for coffee. Presumably he had developed, techniques specifically well suited to the production of cannabinoids. But what sort of happened after that? How did you come up with the name? How did you decide to actually put it together?
So good question. So Jay Kiesling, who's the professor at UC Berkeley, is one of the kind of pioneers and one of the really well-known kind of folks that are, basically engineering yeast cells to make natural products. So his lab was probably really well known for making artemisinin, which is a anti-malarial compound that's used all over the world and was funded by One World Health or basically was funded to make that at cost to decrease and stabilize the price of artemisinin. And so he's always been interested in saying, what are kind of the natural products that exist out there that could be beneficial to people, and how can I use my lab to seed, figure out whether some of these technologies can be used to make these things commercially relevant. And so, around 2016, I think in Jay's lab, it was right around the time that CBD was going through, I think, phase 3 clinical trials in Epidiolex. And Jay, I think, looked at that and said, hey, here's another plant-based molecule that's being used as a pharmaceutical ingredient and, is probably going to be relatively expensive, as most plant products are. And, wouldn't it be interesting to see if we can get yeast to make that, right? And so that's kind of the genesis in his lab. And he had a couple of students and postdocs working on that to say, okay, how do we get yeast to make these products? And they found, they kind of found some of the, one in particular missing enzyme in the pathway that, is the thing that makes CBG or CBGA from the plant. It's a prenyl transferase. And so that was, one of the key discoveries that they made at the time was they could all of a sudden get, call it, hundreds of milligrams of CBG out of a yeast cell, where people before were probably making, a milligram or less, right? So all of a sudden they found this thing that, the way we used to talk about this, or Jay would talk about this sometimes too, is that, what was in the kind of space before was kind of like, called a moped or something like that, scooter, right? And that what they had found was the jet engine that was really required to kind of get yeast to make these things at a high or a commercially interesting level, put it that way. And so that's what he kind of, when Jay had coffee, he said, look, we've found what we think is a missing enzyme here, and think that, cannabinoids in general are, a broad class of compounds that are, very, very interesting and things that need to be studied more and be made more available so we could even study them. And, people will say, well, what got you interested in the cannabinoid space then, or how did you think about this before you made the jump? And for me, it was like, I've heard stories of people, they go to, Amsterdam or other places and go into the coffee houses and talk to people and see, what their experiences are. And that was help them see that there's potential here. And for me, it was like, the National Academies of Sciences had just released like a 500-page report about the potential therapeutic uses of cannabinoids and what, some of the science evidence was for them being effective in those indications. And lo and behold, there's a lot of opportunity here. There's a lot of implications for what these things can do. And there's just more science that needs to get done before these things can come to the market. But it's a huge opportunity to have, positive impact in the world. And why wouldn't you try to jump on that?
Mm-hmm. Do you ever, so when you're doing a lot of the work downstream of the actual cannabinoid production, when you're looking at receptor binding activity and assays like that, do you ever, there might be a kind of conflict, I would imagine, where in academic science, you sort of like get to tell everyone everything. Yep. But in industry, you don't necessarily get to do that. So are there times where, are there discoveries that you guys make that you publish in an open access way or anything like that? So
I would say that there are stuff that we will eventually publish, yes, but we're not at that point yet. And yes, that is frustrating at times. But this is, I think, classic. Like a lot of folks that are in industry feel this way, because oftentimes people in industry, make discoveries or invent technology that then, a couple of years later comes out through academia or other places and gets talked about as like, hey, here's a giant breakthrough that we made. And the companies are like, yeah, we knew about that already. And it's frustrating not to be able to say that.
Yeah.
But that's, the nature is, it's a competitive industry, and, there are lots of people that are trying to do this at this point. And so we're thoughtful about what we say and what we don't say. And yeah, that can be extremely frustrating sometimes, because it's like I always want to err on the side of transparency, because otherwise it comes off as, hey, you're hiding something here. And that never feels good to me. And it's also something that, I don't think benefits, the cannabinoid industry in general. Like the more transparency and openness and clarity that people can provide about what these things can do and how they're safe and where they're effective, I think that's what the general space needs more of anyway.
Mm-hmm. So how did you guys come up with the name Demetrix? What does that mean?
So Jay Kiesling came up with that one. So it was after Demeter, Demeter is the Greek god of the harvest, right? And so, the idea here was that plants make a whole lot of really interesting rare natural products. And wouldn't it be cool to start a company that was focused on making those more available to the world? And it just so happened that the first class of products that looked really interesting were the cannabinoids.
Interesting. So anything, what are you most excited about that's on the horizon? What would you say are the next sort of critical steps for this company?
Yeah. So, recently we announced moving to, full scale commercial production, which is over 100,000 liters. And, getting that working and running and making, so at that kind of scale, companies in this space would be making, kind of metric tons per year of product. That's when you start actually having impacts in the world, right? Everything before that is just, it's the footwork, the hard work that you have to do to get to that point. But that's the point at which, you're starting to make enough product that's going to get make its way into the consumer's hands and actually start having the impact that you want it to have in the world. And that, I think, is, hugely exciting. And it's also a big challenge, right? So getting fermentation scale up to work is a whole like there's, decades and decades of experienced people out there who have, tried to do that. It's hard. It takes a lot of work, and it's something that we're, just really focused on. So getting that to work is really important. And then on the other side of that is both kind of lining up the customers that are going to be able to buy that much product, right? So we want to be able to sell everything we make. And so, finding and getting, those commercial discussions to the point where people are then willing to, sign supply contracts and such to do that offtake is also, one of the big focuses of the company over the next year. And, I think that, speaks to the quality of the strategy of, like, if you can approach companies with some of the basic science of what these things do and what applications they're good for, and provide them real scientific data that says, look, these are what these things are going to be good for, and how they're better than some of the ingredients you're already using today, and how they're more sustainably produced, how they're, all the advantages of fermentation. It makes that commercial approach so much easier and gets them engaged so quickly. And that's kind of, where we are today. And, so from a year from now, when I say, where do we want to be? It's, that manufacturing facility is up and running and humming and doing really well. And we've got people, buying the product, and we've been through, all the safety and regulatory stuff that we need to with the FDA. And we've also, continued to do innovation inside the company to bring, additional cannabinoids up to the point where they're ready to go into that manufacturing facility. Like, that's the thing that's exciting and I look forward to over the next couple of years.
How big are you guys? How many people work here?
So right now we're, over just a little over 60 people.
Okay. Wow.
And so, still, I would say moderately small, but, continue to grow. In 2017, we started off as like 4, then 5, then 11. And so, one of the biggest challenges for any company is growing from 11 to 60 comes with a lot of changes.
Yeah.
And that's, especially people there in the early days, they're used to knowing everything that happens everywhere. And when you're 60 people, you can't do that anymore. And that's, having people let go of some of that and adapt to that is one of the natural challenges that every company that grows has to go through.
What percentage of the employees are like bench scientists, like doing actual
that's a good question.
Science?
So I would say about 40 of the 60 are in, on the science or, manufacturing side of things, which is still a lot of process development. And then, the other 20 are a mix of, and some of those 20, because of what we do involves in the lab, at least, a lot of automation and computing tools. We have a bunch of software engineers that, work both with the R&D team, but they work elsewhere with the manufacturing team and other places as well. So it's a real mix of different disciplines. So even within the R&D team, we've got, people that are yeast geneticists. We've got people who are, what we would call like metabolic engineers, who kind of understand broader physiology of how cells work. We've got, high throughput screening groups that are, how do you measure how much, yeast cells are making of these products in really high throughput. We've got analytical chemists that are there to say, how do we measure in high throughput how much cannabinoid and other products these cells are making. We've got fermentation engineers, downstream purification engineers. There's just a whole broad swath of kind of technical experience that spans biotechnology, automation, and computing. And it's exciting to. And also there's challenges in getting all those disciplines to kind of work together. And that's, one of the things that makes it fun and exciting too.
How much were you guys impacted by COVID?
So personally, I think there's, personal and professionally, right? So personally, COVID changed our lives just dramatically. So, I've got 2 kids, one's 6, one 9, and they, more than a year ago, the school shut down. And then we, basically the California and the Bay Area went into a shelter in place. So shut down everybody, I think on the 17th of 2020. And so we were all then trying to, work from home and teach from home and, do a whole bunch of other stuff at home. and deal with all the stress and concern and what's going to happen here. That was just tremendously unbalancing for everybody. And professionally, we're definitely shut down the labs and shut down everything for a while. And then it was about in the summer of last year, how do you start stuff back up safely? And how our approach has been the people that can work from home and do their work from home have to work from home. The people that can go in the lab and need to do actual lab work and bench work to keep making progress, those are the folks that we want in there. And how do we organize the space, and how do we organize schedules so that we minimize overlap with people and do this in a safe way that everybody in the whole organization feels safe with? Was took time, but we got there, and haven't had any COVID cases inside or transmission inside, and so have been really fortunate from that front. But it definitely has, especially on the science side, has changed. And one of the cool things about science is you're in the lab, there's other people there, you have a cool result, and you can turn to the person right next to you and say, hey, here's a cool result. What do you think this means? And there's like there's like, kicking around of ideas. And that's how a lot of innovation kind of comes about. And when everything's more structured and people aren't in the labs as much as they were before, a lot of that communication like kind of disappeared, and people felt that. It's like, I don't have like that critical mass or those people that I can kick around those ideas with anymore. And so it's been about how do you figure out how to use, Zoom and other tools to kind of like bring that back in a way that people get that, feeling. It's still not quite the same, but, people have found ways. Everything's become more scheduled and kind of like, but all that stuff is still starting to happen and has started happening and is important in pushing the science forward.
Interesting. Well, any final thoughts you want to leave people with, Jeff, before we stop?
Oh, that's a good question. I mean, I think that, what would I say? So I think on a couple fronts, I think it's important. One of the things you touched on, is what advice do you have for people in graduate school or in, as postdocs, or even considering going to graduate school? I mean, science, the training that you get in graduate school and science in general is just super cool. And if it's something that you're interested in, there's so many different areas. Like science is so big and broad that, you will find a place, and you should try it and do it, because it's super rewarding, to be first feeling like you're discovering something for the first time ever, and nobody else in the world knows it, and you're now like a world's expert in something. And that is what graduate school is all about, is making you that world expert in that thing, and teaching you how to, design experiments and, test ideas and get to kind of deepen human understanding of the natural world. And that is super cool. And I would, definitely encourage people to do that. That said, like there's what you do after graduate school is, there's a whole bunch of different opportunities, and people should, try as many of those different things to find the one that works for them. And industry is right for a lot of people, and it's probably not right for everybody. And so try things out and find the place that feels right to you, and that, you get out of bed every day and say, this is cool, I want to keep doing this. On the, cannabinoid side, I'd say, look, what we're trying to do is, take, basically open the world up to the science and opportunity of cannabinoids and say, this is what they are, this is what they can do. They haven't been studied before. Somebody needs to sit down and really do a lot of the hard work of understanding the science. And that's what's going to ultimately let these things have the impact in the world that, a lot of people actually think they can. And it's exciting to be part of that. And we think it's a huge opportunity, and it's, amazing to think about how you can use biotechnology and yeast in particular to make these things more available, and to then use that availability to drive that science forward and eventually make its way into people's hands as things that actually improve their lives. So, for me, what I find rewarding is, science has positive impacts in the world. We've seen it COVID, with COVID vaccines. It's just so much stuff that's science around us, and it's just so exciting, and people should try it.
Awesome. Well, thanks for joining me, Jeff. This was really interesting, and
Absolutely.
I look forward to seeing what you guys come up with the next few years.
Yeah, no, this is a super fun discussion. I really appreciate you taking the time to do this. It's fun.